Quantitative data is numerical data about quantities which can be measured and written down with numbers. Once this numerical data is collected, the researcher uses what is collected or observed into their research. For quantitative data, the information you need is numerical data which is analysed using statistical methods. If there aren't any numbers involved within the research, it can't be quantitative research. There are four main ways of collecting quantitative data, which are field-based experiment, laboratory-based experiment, and observation and questionnaires. This type of data is usually recorded systematically so it can be analysed within a computer database. Also, it can be collected by simple graphs which show the correlations between two or more items. Lastly, you can use a cluster analysis, which is useful for identifying relationships between groups where there is no clear hypothesis. You should collect quantitative data via probability sampling because this type of data is collected at random, and therefore this type of data collection stops out the possibility of sampling bias. Also, you need a large amount of participants for quantitative data because it is essential that your research results are representative, as it is crucially important that when you conduct quantitative research you have a large number of randomly selected participants in each group you survey. This is because the research will then be more representative and generalizable and therefore more reliable, which will make it a better research. Once you have collected the data, you can compare it with other researchers who have done similar studies to see if you got similar results and whether they found anything different to your research. Types of experiment. The two types of experiment I'm studying are pre-experiment and quasi-experiment. Quasi-experiment is an experiment with a random task that ensures that both the experimental and control groups are corresponding. Pre-test is a circumstance that is observed at two time points, one before the research and one after the research. Changes in the outcome of the study are supposed to be the results of the research. Once the research has been completed, there are three essentials of true experimental research that must be measured with a control, randomization, and manipulation. Firstly, the control is there to stop the effect of extraneous variables from influence the outcome of the research. This guarantees that the outcome is caused by the manipulation of the independent variable. Therefore, it is crucial to keep all of the other variables constant. Randomization is the random sampling of respondents into groups. Participants should have the same chance of being sampled into a group in the research. This ensures that the outcome of the study is due to the manipulation of the independent variable. Manipulation is when the researcher will purposefully change the independent variable, which is the condition that will be applied to the experimental groups. Intervention experiments pre and post test should have a very clear testable hypothesis so that there is no objectivity. The researchers know exactly what the research is about. Also, you need to be able to operationalize all concepts so it can be applied to independent variables, dependent variables, or covariables so that they are put into use. Also, it is important to try and control all extraneous variables, even though they are undesirable variables and are very hard to control. Experimental method advantages and disadvantages. The advantages of with this method is that the researcher controls the pace and location, so therefore they will be able to host the research where the respondents have a natural environment, so like somewhere they're used to, which will increase validity because they'll be used to that. Also, this research is representative and can be generalized to the population from which a sample was taken. Finally, if the researcher is able to control the confounding variables, then we will be able to see the cause and effect between the independent and dependent variable. However, the disadvantages are that volunteers overrepresented in the US and the UK, males and university students, so questionable. So this is questionable generalization beyond these participants. Finally, there are some limitations regarding ethics and health and safety, which are all important aspects of this method. Finally, and also, their participants know they're participating in a research, and this may bias reaction and change their behavior because they know they're being researched on, like the Hawthorne effect. Experiments and groups. Intervention experiments should have a very clear testable hypothesis so that there is no objectivity and the researchers know exactly what the research is about. Also, you need to be able to operationalize all concepts so that it can be applied to independent variables, dependent variables, or covariables, so that they are put into use. Also, it is important to try and control all extraneous variables, even though they are undesirable variables and very hard to control. Post-test. The post-test is randomly assigned into groups which are experimental groups and control groups. Neither group is pre-tested before the application of the research, but once the research starts to go underway, it is applied to the experimental group, and the post-test is carried out on both groups to assess the effect of the manipulation. Pre-test is where the respondents are again randomly assigned to either the experimental or control group, but instead both groups are pre-tested for the independent variable. The experimental group receives the treatment, and both groups are post-tested to examine the effects of manipulating the independent variable on the dependent variable. Experimental groups are groups in an experiment that receive variables being tested. Only one variable can be tested at a time to make the test reliable. The experimental group is compared to a control group because it doesn't receive the test variable. Control group is defined as the group in a study that does not receive treatment by the researchers and is then used as a target to measure how the other tested participants do, and is used in an experiment as a way to confirm that your research actually works. Designs. Matched pairs. Matched pairs is when there are two equal groups of participants and each participant is matched with a similar participant in the other group, e.g. they're roughly the same age and the same interest, IQ. Then both groups then take part in a different situation of the independent variable as within their independent groups. This will allow the researcher to control the participant variables that might otherwise affect the results. Independent groups include participants who are only given one condition of the independent variable. There can be several groups of participants, but each group only takes part in one condition of the independent variable and does not repeat anything. This means that the dependent variable will not be influenced by other effects. Crossover designs are observational studies as well as controlled experiments where participants are exposed to more than one treatment, but the second condition could be affected by the first treatment. Repeated measures. Repeated measures uses the same subjects within each part of the research, including the control, and therefore the same participant takes part in each condition of the independent variable, and therefore each condition of the experiment includes the same group of participants. However, this means participant variables are decreased, but it saves time because all of the same participants are used all the time. Experiments. Laboratory experiments are experiments that find investigations and see if a hypothesis is scientifically tested, but in a controlled environment like a laboratory. The advantages with laboratory experiments are you are able to control all of the variables, which makes the test more valid. Also, the equipment is usually top quality, and therefore you can get more accurate and precise results. It has a high internal validity, which allows you to have a high confidence as a researcher. The disadvantages are it doesn't reflect natural performance setting, so it's going to decrease the validity. Also, it's expensive equipment, and you need training to use this equipment. Also, there's a loss of ecological validity and realism to world environment. Field experiments are done in the everyday environment of the participants, like e.g. real life. The advantages with this is it's cheaper and more accessible. It helps to support ecological validity for your research, and involves collecting data in an environment that simulates the one that the sport analysed is being played in, which will increase validity. The disadvantage is that there's less quality equipment than a lab, and also the research may take more time. Finally, difficult controlling confounding variable means lower internal validity and therefore less reliability. National analysis and observations is the study of movement patterns, strategy and tactics in team sports. The advantages with this is that it can show strengths and weaknesses. Also, it can of the participants. Also, it can compare or show improvements. We can compare to other researchers because we have video footage. Also, you can see the video and annotate it later, so it doesn't have to be at that time. The disadvantage